And I will stop there.
All right. Thank you, Dr. Weil. That was really interesting talk. So, all right, I guess we'll move into the Q&A session. We have a ton of questions, so I'll go ahead and look through what we've got. And all right, so I guess I'll start with you, Dr. Rahman. So I'll just go on the more upvoted questions and then work our way down. So the question is, did you look at metabolic functional features that differentiated the effective consortium from an entire fecal homogenate, that is like FMT?
Yes, yes, we did. We found the same sorts of things that we had found with respect to the consortia that were not working, but our interpretation of that is that the synthetic consortium that we have are working in a completely different way than the FMT. The FMT is from a mouse fecal pellet. The strains that we have are from human commensal bacteria. So it's a funny model system in that sense, because the interactions are not necessarily going to be along the same axis. The metabolomics directly correlates with this. The mechanism by which the FMT works is likely more of a reconstitution mechanism. But when we looked at the 16S sort of engraftment statistics of our consortium, what we found was a transient engraftment, and then they all went away. And it correlated with KP going down. So what we think is that the short chain fatty acid production and the depletion of the amino acids are specific to the consortia that we made, not like a global trend for fecal microbial transplant.
Interesting. By the way, I forgot to mention, if either of you two have questions for each other, feel free to jump in. I hopefully, yeah, keep it interactive if you have anything.
I actually do, but that's okay. We can
burning curiosity, so to speak. So, all right, I guess I'll bounce over to you, Dr. Weil. Let's see. Do you think PA and VC interact or have co-evolved in their environmental reservoirs niches in addition to the host? And I kind of talking.
It's a good question. I mean, Paracoccus immunovarians was first described in soil, and that's really where there's really very little known about it. Other than that, it was noted in kind of several different microbiota studies, like population-based studies in Malawi, in South America. So it was known to be part of the gut microbiota, not in Western populations, but in other populations. And it's not very abundant at all, but I think we've seen over time that even things present in very low abundance can have a big effect on the gut microbiota. The low abundance almost steered us away from looking into it further, but it was really, when people have cholera, we've previously shown their microbiota is obviously obliterated with not just the infection, but with the treatment. And there's really not a lot there when people have cholera, but Paracoccus was still present even during cholera. So that was very curious, that this low abundance organism was still there. And now it makes sense why, if they were coexisting in a biofilm during infection, then so that is kind of what caught our eye. But did they co-evolve in their environmental reservoirs? I mean, I have not ever heard of Paracoccus described in the ocean, which is really where Vibrio cholera lives, in low salinity kind of, areas where freshwater and saltwater mix. I don't know that Paracoccus has ever been described in those environments. So I would say probably not. I'm not sure, how they are benefiting from one another, other than that they live together in this environment and have some more protection from having increased biofilm.
All right, interesting. All right, All right, Dr. Rahman, this one is pretty highly upvoted now. I don't know that I mentioned that I was doing them in order of like popularity. A bunch of people jumped in. How can you confirm that microbial strains from 20 different healthy individuals didn't inhibit the growth of each other in the consortia?
I mean, we can't. And it's we can't, and that's kind of the point. In a lot of ways, like we want to assume nothing about what is possibly interacting with each other. All we care about is the ultimate function. In the same way that evolution doesn't care about what interacts with each other, all it cares about is fitness. So our idea was to create willy-nilly, so to speak, I mean, with the constraint of genomic diversity, create lots of different consortia without regard for what eventually would survive, what interacts with what are the dynamics in between, and purely select off of functions. So it's quite possible that actually, you know, if you dig into the communities, there's a bunch of warfare going on between the commensals. And in fact, if if anyone like took a very close look at this synthetic community 15, SynCom 15 that we made, it has both Glaudia and Bacteroides, which are like opposite ends of the spectrum, so to speak, in the quote enterotypes that people describe. So yeah, I think it is entirely plausible that there's warfare going on, but I think that it was consistent with our design principle to not care.
All right, yeah, okay. All right, let's see. Dr. Weil, do you think these microbial sphingolipids are having their immune effects locally on the gut epithelium, or are they going into circulation and impacting distal tissues?
Yeah, it's a good question. Microbial sphingolipids have recently been shown to be absorbed and integrated into the into human lipid metabolism, which is really interesting. That was shown primarily in, I believe it was in like liver and hepatic lipid processing, where tagged lipids that were broken down, originally made by bacteria, were integrated into the host. So I think that it's most likely these are operating at the level of the epithelium, and we are going to test them further in the epithelium to show that using cell culture or enteroid models, in addition to the other immune cells present. So I do think it's more likely happening at that surface. The immune pathway of how innate immune responses impact Vibrio cholerae, I didn't show what we think it is, but it would be that innate immune response at the surface is then stimulating downstream pathways that result in the cognate B cell and T cell interaction in the germinal center, and that this specific interaction that we think is innate in nature is related to function and development and maintenance of mucosal associated invariant T cells and possibly T follicular helper cells. And there are several studies showing that these are involved in the long-term immunity that's made, that then results in these memory B cells that secrete IgA, likely IgA at the surface, that inhibit Vibrio cholerae motility. So I think that it's happening somewhere in there, right? From the germinal center in the lamina propria of the gut is not that far away from the gut epithelium. So I would say distal tissues, probably not. But I think it's happening, in that gut lymphoid system somewhere.
All right, okay. Yeah, interesting. All right, so Dr. Rahman, are there any other tests or targets your team is interested in going after, in particular, like C. diff?
We're actually going after VRE right now, which we have some data on that's pretty positive so far. Yeah, and I mean, I like the spirit of that question, that it's a good one, which is how one, if it's truly a platform, so to speak, then try some other functions. And I think it's a worthwhile thing to do. So it would be nice to march down, a multi-drug resistant pathogen 1 to 2 to 3, and have some mapping about how easy it is to churn through to try to get to a solution. So VRE is next, and then maybe one day C. diff.
Cool. Yeah, I hope we get there. Dr. Weil, have you looked to see whether human-derived sphingolipids induce the same response in macrophages? Is it sphingolipids in general that produce lower baseline inflammation, better vaccine response, or is it specifically microbial-derived sphingolipids?
Yeah, great question. So we have focused on testing sphingolipids, like lipids made by bacteria that are either specific species of those sphingolipids or not. So we focused more on getting to the structure of what is that lipid that is responsible for this, or that ceramide head group that is a class of lipids that are mediating this. And we have not tested human-derived sphingolipids, although there are many of them in stool. And I'm sure, in stool, it's a bit challenging to dissect all of these different antigens and what their effect might be. But we do plan to study human-derived lipids as well, and other lipid types. But we focus mostly on fractionating lipids and really trying to identify what is the effector group or groups that we can test.
All right, bouncing back to you, Dr. Rahman. All right, so this one might be a bit broad, but if you already have a genome, you have access to a rich set of biological mechanistic knowledge on each organism. What if you used more of this information beyond relatedness as a prior in the design pipeline, like you've presented?
Yeah, it's certainly possible to do that. I'll just say one caveat. We don't know much about the genomes of these bugs. Most of the genomes are homology to E. coli K12. And even in that, it's like very, I think we know 70% of the E. coli K12 genome. And then like, if you go to B. subtilis, 30%. And then after that, it's like 2%. Like it's very, very low de novo functional annotation. So a lot of it's based off of homology, which is incomplete. So take that as a caveat. You say you could probably parameterize on KEGG pathways or GO pathways or something else other than what we did was orthologous gene groups, similar to COGS from what or CAGs from what Anna was saying. But I think it's possible as long as you're okay with the annotation scheme. So that's the biggest kind of balance that one runs is, do you go down interpretability or do you go down what, biology would provide you, which is the genome sequence?
All right, Dr. Weil, do you think patterns might be different with Prevotella? Is there epidemiological evidence for differential vaccine response in people with Morobacterides versus Prevotella?
Thanks for the question. I'll just want to comment on the previous question first. That is exactly why we couldn't use CAGs to identify, what strains should we test in vitro, because the annotation is just not reliable. It's just not even close. So that is why we had to create this method of identifying specific strains. It would have been great if we could say, wow, this function is totally related to the immune response. That would have been many steps further, into understanding potential mechanism than needing to identify a strain and then take it to another step. So it's really a weakness, I think, of trying to study the microbiome is the lack of annotation of so many of these anaerobes and other bacteria in the gut. But that is something we definitely, hopefully as a community, can improve over time. So regarding Prevotella, great question. Prevotella, as many of you may know, is a lot harder to culture from frozen stool than Bacteroides. So that's been one of our challenges. We do have a number of isolates, but just not the number we're able to get in Bacteroides, especially when this stool is being frozen in Bangladesh and shipped here. Those isolates are harder to come by, but we are hoping to have a way of isolating more of them in Bangladesh so that we can preserve them better and study Prevotella as well. Prevotella has so much genetic diversity. I'm sure that we would have a lot of things to learn from studying Prevotella as well as Bacteroides. But for the specific question of sphingolipid induced or microbially derived sphingolipids, it would be great to be able to study both of them. And we have a few strains we're working on now.
All right. Excellent. Dr. Rahman, how do you think the functional redundancy that is present in nature, how do you think that will affect the outcome or finding of your work, that redundancy?
I think that, yeah, so it's a good question. This 15-member consortia that we built is probably not a unicorn. Likely, like there are phylogenetic walks that one could probably take that preserve the overall function, but introduce genetic differences, metagenomic differences amongst the consortia that one would build. And so the functional redundancy is probably useful in the sense that there are many possible solutions to one problem. Now, and that's, I think an issue with the way that we built what we built, because we don't include functional redundancy in our consortia design. So it's not like we're saying, well, in case this bug dies, there's another one that will come along and will have the same function. We actually just went into it kind of like just tell me the group to make. I think that if one were to now ask, how do you design translatability? You know, this is one of the questions that I had asked at the end, which was, instead of designing a microbiome for a specific environment, how do you make a microbiome that works across environment 1 through N? You probably do need functional redundancy there, and nature has probably done that for all, you know, our diverse microbiotas. So I think it's a useful thing to keep in mind for future design processes.
All right. Yeah, this is actually kind of a question I have too. Dr. Weil, do you think the work you've done could lead to the design of like a probiotic that could be co-administered alongside vaccination for cholera?
Yeah, that would be the ultimate goal. Using oral cholera vaccines has been really successful in that, you know, you can use them without a cold chain of, you know, needing freezers all the way along. And they're easily administered, you know, they're very convenient, but they just don't work that well, unfortunately, especially in the right groups. So there are people who are looking to study other formulations of vaccines as well, but this is really what we have now, and there's not like a giant pharmaceutical industry trying to make better cholera vaccines, to be honest. So you know, using the ones we have and making them better is a focus. And I think if there was a way to administer a probiotic or you know, even like there's a group of organisms, which many of you are aware, the generally regarded as safe, they're called grass organisms that are, you know, they're not approved necessarily by the FDA, as, you know, most supplements are not, but they're generally regarded as safe, just like the name. They've been used in millions of people. People have taken them as supplements for years. And if an organism like that could be used to augment immune responses, even by a small amount, that would be something that could really change the efficacy, especially in that short term when there's that explosive epidemic that's spreading rapidly. You know, a 2-dose vaccine that has to come from somewhere and then be given multiple doses before, you know, it's just, it would be much more realistic to use something short term for augmenting immune responses or for a prophylactic in the setting of one of those situations. So that's really the ultimate goal of our work, is to see how we can use the gut microbiota to do that. You know, I am also interested in understanding mechanisms, but I think most importantly would be finding something that could be used in humans. And we have other, I'm part of a clinical trial of using antibiotics to prevent cholera in household contacts who are at high risk, but it would be better if we had something else.
All right. Yeah. Oh, that's interesting. Dr. Rahman, yeah, I think I missed this question earlier. Do you expect the suppression function of a community to be related to the phylogenetic diversity of the consortium? Maybe having more strains of a similar taxon is optimally suppressive.
It's possible. There are people who have created, like some of the original work in trying to create defined consortium was done by Kenya Honda a long time ago, 10 years ago, 2013, where he showed that a group of Clostridial strains can upregulate Tregs in the intestine. It's entirely possible that could work. One interesting result that we've seen that I didn't put on the slides is that if you take any one of our members individually and try to see if any one of them work, they don't. They actually all fail miserably. And like there's no difference, I think there's only one member that even does anything to Klebsiella pneumonia in vitro. It's like half an order of magnitude. But then when you mix all 15 together, you get 8 orders of magnitude suppression. So it indicates to me actually that there is some probably cooperative thing happening that's phylogenetic, dependent on the phylogenetic diversity, not on the specific strain. But look, like there's an enormous amount of complexity to try to answer that question, because strain level variation could be important. We would have to assay each one individually, of probably all of the 700 strains that we have. But to a first degree, my gut says that it's probably a cooperative thing across diverse functional groups.
All right, and I think just as a last, my understanding is you, Arjun, have a question for Dr. Weil. Do you want to go ahead and ask? Maybe
I
I'll do the last question.
do. So, well, I had 2. I'll ask one. So you probably know about some of the work with malnutrition in Bangladesh. My question to you was, could you imagine a prebiotic that may help with, like, as an alternative to a probiotic, and which I think is probably a controversial thing to ask, in the sense that we don't think of therapeutic foods as the first go-to. We think of probiotics as the first go-to, but the kinds of access you get from prebiotics are immense. I just wanted to get your thoughts on what could there be foods that could help augment vaccine responses?
Yeah, I think I think it's a great idea. Anything we could do would be something that's safe would be great to be able to use. I don't think it's been studied. It's definitely been studied, especially historically, regarding preventing infection. Like, people know they drink contaminated water and then they eat like a lot of chili peppers, and are they protected? There's even papers about like something similar to that, as far as killing an organism, like once it's already in your body. But for immune responses, I think there's a lot of potential there we don't know about. I mean, a lot of foods are immunogenic. They have antigens that we react to. It makes a lot of sense that by altering the baseline level of, immune activation in the mucosa, and especially, oral vaccines are absorbed in the small intestine. So that's a place where this interaction between antigens from the environment and the gut are happening. And that shapes the platform that we put a vaccine stimulation into. So it makes a lot of sense that would be important. And diet is definitely one of the areas that is under investigation for why people have different vaccine responses in different cultures and countries and genetic backgrounds. Like, diet is definitely on that list, but nothing really definitive has come out about it. And I think some of these interactions are just so complex with human genetics and different foods and the sanitation of the food. I mean, it's really hard, like studying malnutrition and the gut microbiota alone, like just dissecting all those factors it's really a large job. I mean, these data sets in the microbiome world we use are huge, but in humans, the complexity is like so much more variable and sometimes unknown. So I think that area has a lot of potential. I'm a big fan of prebiotic studies as well, but in this field, it hasn't really been looked at extensively yet. Thanks for the question.
All right, well, I think that kind of wraps up our time together. I want to thank you both profusely for agreeing to be a part of this. It was really interesting.